Thanks, James, for inviting me to talk. I'm the director of the Southeastern Cooperative Fish Parasite Disease Lab, and it's known by a lot of names. Some people call it the AU Fish Disease Lab, the Diagnostics Lab, the Co-op, the Auburn Lab, the Disease Lab. So we got a little bit of a branding problem, but basically all of those things are us. I want to give you a little overview of what we call the co-op, because a lot of our stakeholders are in this room. We try to spread the word about the good work that we do. So I'll tell you a little bit about the AU personnel within the co-op, tell you a little bit about our state and federal partners, some exciting infrastructure improvements that are likely coming with our partnership with the service, and some future challenges and opportunities. But James invited me to talk because I got a little grant from the program Cindy was describing, and so that's the title of our grant. And it's really one small piece of a much larger topic, which is the consideration and inclusion of pathogens and parasites in the context of aquatic nuisance species and aquatic invasive species. A lot of the most demonstrable invasive species on a global scale are actually pathogens. There was a virus called COVID that we deal with, which is also a form of invasive species, but get to that later. But well, no, I won't. But anyway, how do I go like that? Yeah. Okay, hot dog. So this is the cooperative. We've got a pretty large footprint geographically, but also in terms of our funding partners. We have annual or 5-year contracts through memoranda of agreement with Alabama, Inland and Marine Resources Divisions, Georgia, the Carolinas, West Virginia, Tennessee. We also do one-off contract work with a lot of other states, Pennsylvania, Virginia, Florida, Mississippi, et cetera. We also have other contracts with the groups listed there, a lot of federal money, including NSF, USDA, NOAA, pretty much every agency that you can get money from, we've tried to get money from and succeeded. We're proud of the fact that, so for the state dollar contributions to the cooperative, that annual contribution that each state makes for us to handle all their hatchery checks, handle their fish kills, consult with their state biologists on aquatic invasive species. For every dollar that we get from states, we get $8 in federal from competitive sources. So we're pretty proud of that. We really try to amplify that impact that we have. This is the disease lab. We've got one of the largest analytical labs on Auburn University campus. We're very fortunate to have really nice facilities. Within our labs, we pretty much can cover any need imaginable in aquatic animal health, including taxonomy, systematics, parasitology, virology, microbiology, pathology, and we're starting to get into some eDNA issues. That's me. I'm a professor in the School of Fisheries. I direct the co-op. I'm also the lab head of the Aquatic Parasitology Lab, which is the research component, the basic research component of our group. Do some editing for some journals. We have $3.5 million in extramural grants and contracts now that I manage. We've got a bunch of people. Those numbers actually aren't updated. There's going to be more people in our group than that, but we have a mix of PhD students, master students, and full-time hard money positions working in the diagnostics lab. I'm not sure why all these are so dark, but anyway, this is Haley Dutton on the left, and she's with us today. Can you just wave? Yeah, she's actually the one that does all the actual work in the lab and manages everything. If you're a client of the diagnostics lab, you deal with her on scheduling and money, and I'm doing my whatever I do. That's Steve Curran. He's a parasitologist. We have 2 master students right now, Haley Knudsen and John Brule. John's working on some neat stuff with invasive carp pathogens. Can you believe that not one parasite has been reported from silver carp in North America? Not a single one. That's how little emphasis is being put on parasites and pathogens of aquatic invasive species, if not for James and Cindy here. These are the PhD students in the lab. They're working on all kinds of stuff from myxozoans to other helminths to viruses. One of the PhD students of note is to the far right there, Justin Kroll. He's a vet that came back for a PhD, and he's actually looking at some viral pathogen, some invasive exotic viruses that infect salmonids and giant tiger prawn, which is kind of neat, and a lot of other things. All right, so in specific, I want to talk about real quick what we were actually funded to do and what we did with that money. It's kind of a long story, but before we discovered the pathogen that causes Salmonid Whirling Disease in the Southeast, the dogma was that parasite magically could not range in the Southeast, even though we have abundant trout resources, cold water resources here. It had never actually been detected in the Southeast. And so actually the first trout that we looked at in the diagnostics lab were infected with the causative agent of whirling disease, which is Myxobolus cerebralis. We also provide the first documentation of whirling disease in wild trout populations in the southeastern United States, and the first evidence of an altered life cycle for the parasite in the southeastern United States. So parasites oftentimes have what we call indirect or complex life cycles, where they require multiple hosts to complete their life cycle and produce progeny. Not all parasites are like that. Some parasites have what we call direct life cycles or simple life cycles, like coronavirus is a simple life cycle. It's transmitted from person to person. It doesn't need a rabbit or a snail or a mollusk or another mollusk to complete its life cycle. It just goes from person to person. M. cerebralis actually has a complex life cycle, so you would think that it would be less likely to be introduced, but that's not necessarily the case. So we published this work back in I can't see anything anymore, so I think that was 17. I don't know, it happened back in the past. We first detected the pathogen in 2016 in North Carolina, and we had some really good partners in the trout program at NC Wildlife, and Jake Rash and Doug Besler, who immediately, once we told them that they were positive for Myxobolus cerebralis, they turned around and gave us a whole lot of money to basically survey all their wild trout populations. They asked me what they should do, and I said, well, if your hatcheries are infected, you need to know which of your wild trout populations are infected, because you obviously don't want to stock infected fish where the trout population isn't already infected. In fact, this is what's happening out west. So the parasite is basically an Eastern European parasite that came in with rainbow trout when they were brought over in the 50s. And it decimated trout populations out west, and there was a huge congressional initiative to figure out how to stop it and whatnot. And then after that died down, pretty much everybody kind of forgot about it, and then we discovered it in the Southeast. A lot of people that were involved in that original problem with Myxobolus cerebralis out west basically were extremely skeptical that it was worth a single dollar to study anything about Myxobolus cerebralis in the southeast. So as usual, the southeast gets the shaft about pretty much everything, especially if it's in freshwater. But the comments were, why do you care about that? It's probably been here for a long time and it hasn't caused a problem. Basically it's nothing to see here. It's not going to be any different than what's going on out west. So anyway, what we did was we pretty much carpet bombed the state of North Carolina with samples. We disproved the first idea that, well, it's probably everywhere. Well, it's not everywhere. It's only in certain streams. So that was a really important fact for NC Wildlife to consider in their stocking program, is that the pathogen is not widespread, but it is established in Western North Carolina. So with this map, we have the positives there. There are the triangles just to show sort of how widespread it is, but the fact that some streams remain negative, and this is after repeated sampling across seasons and across years. So that was useful information for them, and they've basically restructured their trout stocking program to sort of reflect some of this epidemiological data. The life cycle of M. cerebralis, as I was mentioning before, includes an oligochaete. This is a pretty bad, I need to make a better slide of this, but Oligochaetes, we all know what those are. They're abundant in streams, especially in low flow. When you have a lot of high sedimentation, you get really high oligochaete populations. Oligochaetes are the vector for Myxobolus cerebralis, so there's a correlation between the abundance of oligochaetes and the density of oligochaetes and the incidence and prevalence of M. cerebralis infection in trout. And we've done some work to show that as well. I don't have time to talk about it. Here's some oligochaetes here. Does anybody know how to identify oligochaetes? Yeah, very carefully. Yeah, there's basically like maybe one or two people in the world that can actually identify these with confidence. There's a big difference between using a published taxonomic key that's in a book or an old book versus a taxonomic expert who can maintain the current literature and understand the molecular and morphological systematics of a group. And basically all those people have died. So we're training somebody in my lab to become the world's next expert on oligochaete taxonomy and systematics. But anyway, this is the vector for M. cerebralis. Steve Sepka is the doctoral student that I'm referring to. This is mainly his work, and of course in collaboration with Jay Grash and myself. We sampled, and this is part of what we used the Gulf States Marine Fisheries Commission money for. We started to look at which species of oligochaetes were vectoring Myxobolus cerebralis in the southeast. If you look in the literature, everybody says the only vector is this worm called Tubifex. Well, if you go into GenBank, which is filled with errors and filled with problems, it's a minefield of error cascades and improper taxonomic identifications. If you just pull out the sequences that are Tubifex, what you see very clearly is that a lot of them have been misidentified. So that's a real big problem with GenBank, which is the world's sort of repository for DNA sequences, is that if the investigator who sequenced it and deposited the sequence in GenBank didn't properly identify the organism that they sequenced, you're going down a real bad road there. And different people use those sequences to test hypotheses, but if the sequences are misidentified, then it's a great big mess. And what we realized is that in the southeast, not only is what's formerly called Tubifex a massive species complex, and a lot of sequences have been misidentified, we actually confirmed that the infections that we have in the southeast are not Tubifex. In fact, it's a completely unrelated oligochaete. So those naysayers that had experienced the problems out west with whirling disease and salmonid population declines were wrong. And in the southeast, the parasites actually adapted its life cycle, or maybe it was here all along, and when it got shipped out west, it acquired Tubifex as a host. We don't know, because a lot of times when invasive pathogens when you finally detect them, they're actually already established. But anyway, so this was really interesting work, and it really helped provide a tool for NC Wildlife and the other agencies that we've helped with whirling disease, including Georgia, South Carolina, and West Virginia and Tennessee. Oh, and come to find out, pretty much every state in the Southeast has Myxobolus cerebralis. So kind of funny when you look for it with a molecular tool that has a high diagnostic sensitivity, you tend to find things. So it went from it's nowhere to it's in every state. Which is not unusual for the story or the arc of an invasive species. You know, everybody says, oh, that's not here. And then it's like, oh, yeah, it is. Oh, yeah, and it's everywhere. So we had that same experience. But by doing the oligochaete work, we were able to help develop a non-lethal sampling method for the states to actually survey a stream and know if it harbors the pathogen. Because you don't have to kill fish. You can sample the oligochaetes and know if the vector is positive. If the vector is positive, then the fish that are there are infected. And this is useful too, especially in brook trout populations that are really low in number, and you don't want to go in and just lethally sample all the brook trout that you're trying to conserve. You know, brook trout, of course, is the only native salmonid in the southeast. Although interestingly, I was working with Bernie Kahutska up at the Tennessee Aquarium Conservation Institute, and he just told me about some Oncorhynchus species that in Mexico that are Atlantic Basin native Oncorhynchus species that are undescribed, which is really neat. So we want to actually sample those too eventually. But anyway, so much neat stuff going on. But anyway, this is what whirling disease looks like. We also documented the expression of the disease in salmonids in the southeastern United States. So this is classic textbook whirling disease. And you can see why they call it whirling disease. Interestingly, the parasite stage that infects the fish asexually divides in the cartilage and bone of trout. When young fish are infected, as that cartilage and bone is developing and being laid down, the parasite is also multiplying, and so it's like a tumor that grows right where bone is forming. So you basically get these malformed osteological features in infected and symptomatic trout, and that leads to neurological dysfunction. And because the coloration and the chromatophores of fish are also related to neurological function, you can have this bicolor where the posterior end is pale and the front is dark because the chromatophores are not communicating. And you also get this kink tail, different deformities. So it's a pretty gnarly disease. But you know, the fish still pretty much look okay. They don't go off feed and it's not lethal. But you know, you don't want to be releasing fish like this into a stream because, you know, thank God for Facebook and Twitter and all these wonderful things that make state agency biologists' jobs much, much easier. I'm being facetious. So this is where all the reports start coming in. So, you know, we were told by people, you know, that had done some stuff with this, oh, it's never going to cause disease. You're never going to see expression of whirling disease in the Southeast. Well, here we go. So here's pictures. These are like stuff from anglers. It's kind of funny too, just the way human people are. You know, an angler will see something like this and they'll know it's important enough to take a picture of it, but they won't keep the fish. So we can't ever confirm what caused this, but it's very likely that these are clinical whirling disease fish from the region. Just more, you know, whirling disease fish from wild trout populations, et cetera. There's that bicoloration that I mentioned. It's also kind of neat when you're at the hatchery, you can actually spot the fish that are infected. This is pretty rare for a pathogen, but it's a behavior. One of the clinical manifestations is a behavioral modification of that whirling disease. So you can actually, the guys at the hatchery actually can pick out symptomatic fish. So they took us right to them. You can see, yeah, up in the corner there. And it's kind of a fun game to see how many clinical fish you can pick out. But that one right there that I'm pointing to, bang, got it. Okay. Okay, so we picked up a bunch of those. We documented this. You know, good to know. All this is just really good information for the state agencies and trout managers, because it's a real paradigm shift to realize that these things could be exerting population level effects in wild fish. And also raises alarm among anglers. And of course, who are anglers going to blame in a system where most of the fish that are captured are stocked? They're going to blame the state agency. So they're going to say, why are y'all stocking these sick, deformed, dumb-looking fish? You know, so not good. We published all this stuff, and that basically comes to the conclusion of what we were funded to do with the Gulf States Marine Fisheries Commission report. So essentially, we overturned every assumption about this pathogen in the southeastern United States. It's pretty rare that can happen. So it's not everywhere. It's a distinct life cycle in the southeast. All of these things need further study. There's a lot of related species that I'm going to talk about very briefly in the 10 minutes that I have left. These other related species are morphologically almost identical to Myxobolus cerebralis, but even though they almost look exactly like Myxobolus cerebralis, they don't infect the same fish and they don't cause whirling disease, because these myxozoan parasites are actually very specific to a particular tissue in their host. A lot of parasites just infect the gut of a certain kind of fish, like a trout or a carp, but myxozoans are really neat in that they're intracellular and intercellular parasites that are very specific about the tissue that they infect. And cerebralis targets cartilage and bone, and that's why it causes whirling disease. And of course, who cares about this? Everybody cares about this. This affects, like, these kinds of things affect everybody involved in aquatic conservation across the board. So it's there's a lot of work to go around. And of course, the people that are, hit directly in the pocketbook by this are private fish producers. As more and more much needed regulation is applied to the movement and translocation of fishes across state lines and also, even across river basins, they're under obligation to have their fish checked for diseases, and if they're positive, they can't move them or sell them. Now, different states have different degrees of enforcement of that, and I think that that's kind of a clown show in and of itself. I mean, there's very little interstate regulation of anything regarding pathogens. It's just a hard nut to crack, but down the road, that's going to be one of the biggest challenges, I think, for the fish chiefs and the regulators in the Southeast is how to help people move past knowing that they have a parasite or a pathogen in their fish stock. So that's a big issue, because if you don't give people a way to fix the problem or get around it or somehow work with it, they're going to just avoid testing at all costs, and that's what you don't want. You want everybody to feel good about getting tested, because then we know what we've got. But if the penalties are so high and there's no way to mitigate it, why would you get tested? I mean, that's just poor. Quite frankly, just to be blunt about it, it's poor business practices. If I was the CEO of like some massive trout farm, I wouldn't want to test my fish, because as of now, the rules are super confusing and there's really no incentive to do it. So that's a big issue that I think the people that get paid to think about these things need to think more about. I mentioned brook trout, so I just want to segue now into a few more things about myxozoans, and this is just sort of like a platter of examples of why disease diagnostics and pathology and fish health matters from a regulatory perspective and from a disease diagnostics perspective. So here's a beautiful brook trout, the only native salmonid in the southeastern U.S. It has I mentioned it earlier, it has a species of Myxobolus that we discovered. It's a new species, nobody had ever seen it before we described it. It infects brook trout, and we suspect that records in the literature of Myxobolus cerebralis from brook trout were actually misidentifications of this parasite, which we named Neurofontinalis. So this is Myxobolus neurofontinalis. It's very, very similar to Myxobolus cerebralis, and if you would not use a molecular marker to confirm this as cerebralis, you would think with microscopy that this was cerebralis. So it's a good example of a myxozoan that could lead to a false positive and lead the agency to waste a lot of money and destroy a lot of perfectly, fine fish that are not infected with cerebralis. This is likely a myxozoan that co-evolved with brook trout and has been happily infecting brook trout for gajillions of years, like so many other parasites. And again, parasites really only cause disease when it's a mismatch, when they see a host with no innate resistance, or when a host with no resistance sees a parasite that it's never seen before in an evolutionary context. That's when you get disease. Most parasite-host relationships evolve to a state of benign coexistence, but that's not always the case with invasive species. I'm going to skip over that. So that's the new species we described. On the threatened and endangered species side and just the non-game sort of side of things, myxozoans are also an issue. These were some fish that the water department in Georgia was concerned about, because they were told that these fish had tumors, and they were working with basically the equivalent of the Department of Water Quality and all these things, and chemists and whatnot to try to figure out, okay, we've got tumors on fish, what the heck's going on there? Well, they're not tumors, they're myxozoans. So myxozoans, you know, this is related to the thing that causes whirling disease. They can cause these cysts that look like tumors. So false positive on a fish tumor there. More myxozoans. We've also got some myxozoans that are problematic. This was work that we did with the U.S. Fish and Wildlife Service and Brian Hickson at the Warm Springs Lab. This is the soon-to-be listed sicklefin red horse. It had some tumor-like growths on the skin that they were concerned about. They were trying to bring animals in for broodstock, and they didn't want to contaminate their culture facilities, so we had to look at those. And guess what? It's a new species of myxozoa. So this would also lead to a false positive for Myxobolus cerebralis, but if you nerd out on these things and look really, really close at them, like to me, those look nothing like cerebralis, but it's an acquired taste. We've also got issues with gill lice. We've published all that information, and we're working with the state agencies that know they're positive to sort of mitigate the spread of that. This is a really obvious infection that you do not need a microscope to see. So these are gill maggots or gill lice. These are parasitic copepods, and they cause gill clubbing and some disease in young rainbow trout especially. So they are an exotic invasive species. We don't know the impacts of these gill lice on other native fishes, so we're looking into that too. Don't have time to talk about any of that. Okay, on the virus front, we're all so this is kind of a funny story. So I applied for a grant. I asked James if it'd be relevant to study some fish virus work, and I kind of hastily put that proposal together, so it probably didn't deserve to be funded. But pretty much like 2 weeks after we submitted that, and it was not selected for funding, we started kind of running across some problematic viruses in some of these fish. We're using genetic approaches and conventional cell culture approaches, but there's going to be some neat stuff coming around about exotic invasive fish viruses, and these are basically undersurveyed and undersampled, and we're developing some new probes and molecular approaches to sort of detect these things. Some of these things are OIE reportable pathogens, which means that as a diagnostician, I'm supposed to report these, but that's the long convoluted story, but anyway. We've also done some work with developing treatment protocols for killing things like gill lice on things like striped bass. This was some work we did with the Georgia DNR, trying to figure out how to knock out gill lice as they were hauling broodstock from one river basin to another. So that was fun. We used a trout hauling truck for the different chemical treatments, and got some interesting results. This thing's just stopped changing. Oh. And a lot of this was done by the side of the river. Here are the parasitic copepods that have been doused in chemicals and are dying and suffering. So it worked. Anyway, do I have time for this? Yeah, I'll do one more. So if I can go back. Okay, so social media and fish kills. Not a good combo. Riverkeeper groups are great, but I think the tendency is for some of them not to appreciate how much they don't know. And as scientists, I think that's what we should always focus on, is how much we don't know rather than try to promote how much we do know. It's very easy for some of these riverkeeper groups to have good intentions, but to promulgate really erroneous and misleading information on social media. This was a kill on the Savannah River that had nothing to do with what the riverkeeper people were saying that it did. They went to a local hospital and worked with a medical doctor to isolate a bacterium that's ubiquitous and always isolated from fish, that's very rarely known to be a human pathogen. So all of a sudden the trout are dying because of a human pathogen. So we have to shut down the river and call out the the fish police or whatever, I don't know. But anyway, we helped them diagnose the problem. It's actually a very common problem called red sore disease, which has been known in the southeastern United States since the beginning of time. It's caused by a ciliate, a stalked ciliate, this is it, and a bacterium called Aeromonas hydrophila. When the ciliate and the bacterium coexist on the skin of fish, they cause red sores, and the fish get over them when it cools off, and it's not really that big of a deal, but people report them when they see them floating in the river. So anyway, turns out that that strain of Aeromonas is most closely related to a strain of Aeromonas hydrophila from Wuhan, China. So either we gave it to them or they gave it to us. We don't know. But anyway. Okay, and then just lastly, because I'm out of time, we did some neat work with Bernie and the guys up at the Tennessee Aquarium Conservation Institute. They were concerned about these parasites on an endangered species, the laurel dace. This is actually a bird parasite that uses the fish as an intermediate host. The parasite's called yellow grub. It gets these intense infections here. So Haley and I went up there and worked with them in these streams where the laurel dace are. Of course, you can see the laurel bushes there. And it turns out that the incidence of this parasite infection in laurel dace seems to be correlated with the incidence of tomato farming and bad soil erosion. So we're sort of getting into that a little bit, but we think that when the soil erodes, it offers habitat for a phycid snail, which actually vectors this parasite. And then when the fish gets eaten by a bird, the parasite matures in a bird, but we're sort of looking into that with them. It's a neat sort of human dimensions, farming, land use, infectious disease, invasive parasite issue that's interesting. Okay, we've also done stuff with freshwater mussels, but that ain't happening. I wanted to, I don't know if Alan wants to, you want to take it from here, Alan? I'm ambushing Alan here, I didn't ask him. But Alan and I at the service, we've been working a lot with Brian Hickson at the Warm Springs Fish Health Lab. Brian and our close colleagues, and Alan, of course, and I have been working together on bringing some money for fish health to the southeastern United States. We were able to get, well, go ahead, Alan. Do you want? We're able to get some money in the bill that's about to get passed, hopefully. So it's going to help out Bryan's Center, and it's going to help out Auburn.
In the 22 omnibus bill, there's $5 million that senators shall be put in for fish health across the southeast, with half going to Auburn and half to support our fish health center work at Warm Springs.
This conference will now be recorded.
So it's in there.
So it's in there. So we hope it passes. If that gets passed, we'll have a lot more resources to help state agency biologists without having formal agreements or contracts or anything like that. We're also, just one last thing, I always talk long. Can anybody guess what that is? It's not a coffee machine. It's an eDNA sampler. Yeah. So Smith Root has come out with this product. It's $1,200, and it's have you seen this? Yeah. Okay. And it's an eDNA sampler. So I'm behind on everything, so I just learned about this. But anyway, I show it here because as part of another process, Alan and I are looking for resources to establish a little bit of an eDNA lab at Auburn to fold that into the fish health lab. So it's kind of interesting. And clearly Smith Root is anticipating the remote control not working. Anyway, the next slide basically shows you what the thing is called, and it's called the citizen scientist. So that could potentially be extremely bad. I'm just going to be the one that says it, okay? I'll just leave it at that. But anyway, Smith Root sees that this technology is expanding, so there's a big need for taxonomic experts to get involved in the primary generation of eDNA data to go into databases like Matt was saying. And I was going to rant about the National Aquatic Animal Health Plan, but I'll just skip that. So anyway, sorry I went over by a couple minutes, but appreciate it. Thanks. That's it.
It was worth the extra minute. Thanks. I appreciate it. That was a lot of, that's really cool stuff. So appreciate it. I think it was a good use of the money, honestly. But any questions? Actually, I did have a question. It seems like from what you're saying, and maybe you mentioned this, obviously you're testing these trout. They don't maybe display the symptoms. I mean, but if you did have one that was infected, and you infected it, how long would it take to start displaying symptoms?
It would depend on the age that the fish was infected. Young of the year rainbow trout are the canary. They're the most highly susceptible to primary infection by the pathogen. And basically the dose makes the poison. So if the fish gets a really, really heavy dose of the dispersal stage, the infective stage of the parasite, and environmental conditions are right for the fish to really produce a lot of these asexual stages, it can happen pretty quick in a matter of weeks. But what's interesting about whirling disease is that even after the fish clears the pathogen, because the pathological changes are osteological, they're bone-based, they still look infected even though they're not infected. So it's neat from an invasive species and sort of biological control perspective is that you've got a fish that looks infected that ain't anymore. So it's a great, I talk about cerebralis a lot because it challenges a lot of the conventional notions about like biosecurity and how we should handle these things, but it's a good question. And actually for the Southeastern United States, we do not have that information. So I should say I'm basing that on what's been published, which is all from out west, basically. So.
Thanks.
Yeah.